The demise of cells is not always accidental. In fact, much dying of cells is by design, a predestined stage in development. As you can imagine, this kind of programmed cell death, or apoptosis, is tightly controlled. Non-dividing cells are arrested in G0, the diploid state, before entering another S phase before their DNA can be replicated again. In fact, most mature differentiated cells stop dividing altogether after differentiation. So cells don't divide indefinitely, but they also don't live forever. When they die, they can be replaced by stem cells. As an example, our red blood cells function for about 60 days before they're then removed by the spleen. They're constantly replaced by stem cells in our bone marrow. As for the old saying that death is a part of life, that's nowhere more true than in the cells of an embryo. Here is an analogy that might be useful if you have ever seen workers pouring a cement sidewalk. First, a set of wooden forms is laid out in the pattern of the eventual cement squares of the sidewalk. Next, cement is poured into each of the forms and allowed to set, and after the cement has hardened, the forms are removed and the sidewalk is finished. Think of the wood forms as embryonic cells that were created in the embryo to guide the formation and location of subsequent cells in the embryo. Once their job is done, however, these cells have to undergo a programmed cell death. In other words, apoptosis. Another example of programmed cell death, perhaps one that's familiar, is metamorphosis, in which tadpole or caterpillar cells die to be replaced by frog or butterfly cells. Like many other regulated cell activities, apoptosis starts with an external signal. This diagram is shorthand for a signal transduction pathway that generates a transcription factor. The transcription factor, in this instance the yellow triangle here, activates BCL2 genes that lead to the synthesis of BCL2 proteins. For the frog, metamorphosis results from the activation of genes initially stimulated by, of all things, thyroid hormone, which actually gets inside the cell to deliver its message. Now, let's look at how some BCL proteins work. The BCL2 family of proteins includes Bax and Bak. These two proteins interact with mitochondria to form a channel through which cytochrome C can diffuse out of the mitochondrion. Recall that cytochrome C is part of the electron transport chain, oxidizing carbohydrates and fatty acids in mitochondria. After leaving the mitochondrion, the cytochrome C combines with a cytoplasmic adapter protein, shown here as the green notched box in this cartoon, and the activated adapter proteins now associate with each other and with multiple procapsase proteins. The procapsase proteins are inactive proteolytic enzymes. These proteins are then hydrolyzed themselves, releasing active capsases. Capsases X, Y, and Z are enzymes that now catalyze the hydrolysis of cellular proteins, leading to death by apoptosis. Okay, cells often die because they are programmed to die. What about the unanticipated or accidental or unprogrammed death of cells, say from injury or illness? How can we tell the difference? Well, non-apoptotic cell death is called necrosis, and is characterized by the disintegration of the cell, the lysis, the spewing out of the contents, as shown in the micrograph on the left. Apoptotic cells don't lyse, but instead they usually shrink and contract, and their contents become more concentrated, and the cells themselves can eventually be found engulfed in phagocytic cells, where their molecular components are going to be recycled for their nutrient content.